Hey everyone, my name is Azim. Today we'll be looking at different tissues under different section types. So when we cut tissue, we can cut samples in one of two ways. There can be a cross section, which is a lot like a horizontal or transverse section on a full body, or we can do a longitudinal section, which is where you cut something long ways. So think as cross section as the hamburger cut. Think as longitudinal section as the hot dog cut. Hamburger is, a rounder cut, and longitudinal hot dog is longer. So first we're going to take a look at squamous epithelium. Squamous cells, if you're familiar, the word squamous is referring to flat cells. Think of it like a pancake. If you look at a pancake top down, it looks like a nice round circle. But if you look at a pancake from the side, that would be longitudinal, it would look like a really flat line. The same is true for a squamous cheek cell. So we're going to look at a group of cheek cells here first. We'll take a look at what section they're in. So this is a cheek cell smear, squamous epithelium cheek cell smear. It's not in focus yet, obviously, so I'm going to bring it into focus. It's already been centered. Here we are at scanning objective 40x total magnification, and we can start to see a purple-ish blob in the center. So that is actually a group of cells. It's not just one cell. I know it can get really confusing, especially with something like this, that it's just a small group of cells and there's not much else on the slide. If you go too far, if you zoom in too far, you see this other stuff. That's not the slide. That's not our sample. That's some other stuff on the slide. So that's too far. You want to look for the stained stuff. We stain it for a reason, so it's easier to find. Okay, so that's in focus. Out of focus, too far, in focus. And there's some other black debris on there too. Okay, so now that we're at scanning, we're going to go to low power, 10x objective, total 100x. It's a little blurry. Now it's in focus. Great. Move that up a little. Now we can start to see the nuclei a bit more distinctly, but we still want to get a bit closer. So switching to high power, 40x objective, total 100 or 400x, excuse me. Great, and now we can see our cells. Let me point at a nice distinct one. I like this one, so I'm pointing at it right now. There, I'm putting the arrow to the edge of the plasma membrane, and then now I'm going to move the arrow to the nucleus. Whoops, too far. That's another nucleus, so that works too, I guess. But these are entire cells. All these that you see are dark nuclei and light-colored cytosol and plasma membrane. Because these look nice and flat already, nice and round, this is in a cross section. Back to low power, back to scanning. We're going to switch now, so I just showed you squamous cells in cross section. We're going to switch now to longitudinal section. This is cells from a kidney. So here we are in the kidney. You see a bunch of things going on here, lots of tubes, and we'll talk about what the kidney's made of later, but we're going to zoom in on those weird circle-looking things with the white space in the outline. So here we are at 100x total. Still need to get closer. Here we are at 400x total. All right, so which ones are the squamous cells? Since we're looking for longitudinal cut cells, we need to look for thin, flat cells. Not the ones that look like pancakes like before, like from a top-down view. We want the side view. Where I'm pointing my arrow right now, that is a nucleus of a very, very thin, flat cell. All of this is just one cell. It's lining the white space. This white space is a lumen. So this is the nucleus where the arrow is right now. Here's the membrane, very, very thin and flat. And this type of cell is what's surrounding this entire circular structure. It's called the Bowman's capsule. It's found in the kidney. It's what catches all the filtrate from blood, but we'll get to that later. This is a very thin, simple squamous cell. And a bunch of these cells, as you can hopefully see, I can't point it out easily, are surrounding that entire circular area. That's a squamous cell, cross-section versus longitudinal section. Let's look at one more thing. Hair is a much simpler thing to imagine. When you look at hair, you can look at it from a side view and it looks really long, or you can look at it from a top-down view where it looks crossed. So let's look at the side view, the one that you're probably more familiar with. Here's skin at scanning objective, and here, what I'm pointing out with the arrow, that is where a hair follicle would be. This is the hair follicle. That is a longitudinal cut of hair. But if we look at a horizontal cross-sectional cut of skin, we can find something different. Here we see a bunch of circles. Let me go a little closer to low power. And all those circles are also hair follicles, but they're just cut differently. So this is something you'll need to pay attention to. How are your cells or structures cut? Are they rounder, meaning, well, actually that's not the best instance. If it is an oblong shape, does it look like it's head-on like this? This would be a cross-section, or is the long view longitudinal section? Hamburger or hot dog?